question. And okay, thank you. And conversion to cusp markers, because last year within IT we detected some QTL validation conversion to markers. So we need to validate and convert this into cusp markers to be routinely utilized in the breeding program. I cannot see the people who are attending, and then if they're really from our TAC members to contribute in how we can proceed with the QTL validation so that can help us to achieve our result in fast. We're also trying to in vivo methods where we can test those QTLs and the other population to see if it is working in the same pattern, but they have some more and fast and efficient way, so that we need to take it into consideration. I welcome that comment. We are also in IT also thinking for QTL detection. We have genotyping in the data from our 2020 trials. So to go for more QTL detection for using linkage analysis, and maybe also apply Jivas for the existing data. So we need to use our data mining. And more specifically, I think we are looking for QTL detection for flower sex and cross-compatibility. From our retrospective analysis on all the parents used in the crossing program, we have some information about cross-compatibility index. And we have also the known male and the female parents. And we have some genotypic data where we assess the level of heterozygosity of the parents and the genetic distance between the parents, how that affect the cross-compatibility. So that's is part of the it's Mundo, who is really excellent doing in this work. So we want to go for the QTL detection, combining flower sex and cross-compatibility issues. Also, I will also try to apply the QC and quality control and assurance marker kits that we have about 99 markers. We sent for the validation about over 250 genotypes representing the species. Once the validation is done, then we need to use that for tracking the genetic purity of our materials in the different testing stages, and also for the hybridity tests in the seedling nurseries, at least to confirm those true hybrids to proceed with the selection process. So we also expect more information and markers for flower sex determination from IBRC. I don't know if Professor Terauchi is connected. Maybe we want to hear from him. Hope also he will, during this presentation, he can also comment on that. And we think also genotyping of new breeding lines from the early generation, which we have not been genotyped before, to include in the genotyping using the DATSEC LED, which we have negotiated with EIB. So this is the plan. And for the robust analytical tool and platform for predictive breeding and decision support available for the young breeding. So we are thinking about refine we did a preliminary genomic prediction model. We also applied this prediction in some of our breeding work in IT, but we want to come up with, refine this prediction model with the data we have now, more data, and then apply for ISL for best parent in terms of their genetic value, and also for prediction for clones that with varietal value. That's what we want to focus on 2021. And really we also look forward some really solid support from GHI in this area, because they can assist or help us with this, and also BTI. The other one is the best genomic prediction tools made available in YAM base for use by breeding programs. So I leave this one for the BTI to work this and come up with some tool. The prediction can be integrated into YAM base and applied, because we have the genotypic data, and also we have phenotypic data. And then with the mixed model analysis, if we integrate the genomic prediction model that works good for YAM, I think that can be used in YAM base. The other one is in the project document, we also promised or committed for development of to develop a variety catalog in YAM base to showcase that the best clones are available from breeding programs and integrate with the ability to order seed from the programs. So I've been discussing with Lukas, and then I think they are working on it so that at least those released and the very advanced clones, we took some pictures of different stage, and then the data is there, and we have also more data, so that I have a catalog section in the YAM base that help us those varieties which have been released, which country, which local name, and also if they are new ones and their values. And then if somebody interested from somewhere else, they can also make online selection and then order to get planting material of this. So hope this will be a part of 2021. And then this is all work of BTI. And now the third is new and emerging phenotyping tools and the platforms for agronomic and quality traits, validated and adapted breeding programs. So the plan is, yesterday we have seen they are very interesting and fascinating tools and protocols to capture the quality of yam for food. So we are planning to those tools be available, validated and made available for breeding screening. That's for more of Dr. Busi and the team. And maybe also the RTB food can also contribute on this. We want to really get some ready to go tools and techniques to be applied in large scale screening of our materials. And if there are some associated skills or some tools need to be combined with those SOPs and that's techniques, if that may include training or some infrastructure, if that made available to breeding program to utilize, because this for yam, the food quality is key. You know, it's the key factor determining the acceptability of the products coming from the breeding program. In addition to that, we also faced some, I think it's part of the project document we already committed. So robust phenotyping tool for fast and accurate assessment of large-scale screening of the breeding lines for reaction to pests and diseases. I know from Dr. Lava and the team, they have some tools, but we feel that these tools has to be at least exposed to the breeding program and made available to us. Not only made available, I think I want them to walk a mile to help the breeding program to implement and then use this for screening of their for the breeding population. Maybe we can start at the same for the food quality, maybe include more training or more if you had some infrastructure needed, and also their support being part of the breeding program to assist the IET and also the nurse so that we can increase our efficiency. Because the tools are there, then we need to utilize them in the large scale screening. And also that will help for them to get the feedback for the maybe next remaining year to improve it. The third flagship for product is the next generation of superior varieties or farmers or end user preferred varieties. So the intermediate outcome is the product profiles, breeding pipelines, and the schemes optimized in national and international yam breeding programs. So the plan for 2021 is finalize the market segment characterization for yam breeding West Africa. Hope Bish will be leading this one and help us to get this done, made available for the breeding program. At least we have some clarity on clear focus what our market segment we are addressing. So based on that market segment, then we are thinking of refining the product profile for yam using the result of stated preference survey. This stated preference survey is, I think, is voluntarily supported by Tesfa Michael, a socioeconomist. So we want to finalize the questionnaire and then also sampling strategy. I really want to also get the partners, since we share the survey questionnaire, so that they can comment on that, and then we have a common questionnaire. And then we're also thinking of respondent training. No, enumerators or something training. Enumerators training, maybe between before we conduct the survey, so that we have common understanding of what data what's the expectation, what we are going to collect, who we are going to involve, and then how we are going to analyze, and how that can be monitored centrally and also within the partner, so that at least we get some good data that can help us to refine our product profiles. So we are hoping to conduct this now in June. We are also thinking of maybe the stated preference for stratified by producer. We cannot go the same set of questionnaire for producers, processors, and marketers, but since the processors and the marketers may be small, we focus much more on producers, and then we need to generate some questionnaire and ask them, and then come up with really what is the product profile that we are going to use in the breeding program. And then also under this product profile, we have also the trial cost characterized per program across stage of the breeding process. So here we are, there is a plan with the modern plant breeding project that's with Bayer. We have a work stream team working on it, tracking operational cost in breeding. So we define the standard unit for the cost. What is our standard unit for different stage? In nursery, what's the standard unit? In the field, what is the standard unit? In the lab, what is the standard units? And then how we can estimate the cost for that. So this already initiated an ITA and supported by Bayer under this project. We draft cost estimate for ITA by end of July 2021. And then using that information to extend to the nurse partners, so that they can use that methodology to get their also cost, and hope it will be also. And the Agri is leading this from the YAM part. Agri and Alex are the one who are working on it. And again, for the product profile breeding scheme, we have quantitative optimized breeding pipeline for the YAM programs. That's one of the milestone deliverables. Yeah, so we continue optimization exercise with EIB. Now we are supplying them more of genotypic and phenotypic data, so that simulation also can use the current data set we have. So far we did for the number of parents to use at one cohort of the recycling or crosses, making crosses. And we have also some prediction for the, I think the genomic GPLAP also seems superior as compared to the other, so we'll work on it. And then the remaining we are under discussion is cross prediction. So if we also do cross prediction, optimum testing environment, optimum environment for early generation screening and advanced stage testing, capturing the YAM, TPE in the market segments, and optimum progenies per cross, And also we are expecting the optimum plot size. It looks too much, but hope if we can get this push on that. And then introduce these optimization results to the nurse. I think among those, the seminars, so that once we get those results, maybe we also strategize how those can be applied in nurse program. And we also looking at agronomic and hormonal manipulation of the flowering in white yam and assessment of optimum pollination time in white and water yam. Maybe Mundo, if you are here, you can just, one of the PhD students working with us is with the key area. Maybe I can give him 2 minutes so that he can just explain what he wants to do in this.
Thank you, Dr. Asrat, for this opportunity. Mondo Balama, I'm a PhD student from DRC, Congo. I'm here around, it's about 10 months now, and I've been working on flowering and pollination efficiency in yam. So we are trying to see if there is any way of improving the pollination and flowering in yam. But to put a background, yam is challenged by many factors. Among them, there is that asynchronous flowering. There is low or absent, so most of the popular varieties are not flowering at all. But there is also that unbalance between the males and females ratio, the ratio in terms of flowering. But even the flowering genotype, there is low viability of pollens. And even when the viability is okay, there is also that cross-compatibility barriers among species. And what Dr. Asrat presented yesterday, we found that based on historical data that there are additional challenges as well, such as the lack of skill from technicians involved in pollination. But also there is the problem of weather. So the sites, some of the sites where breeding is being done are not conducive to high success rates for pollination. So that is the background. And what we thought, so based on the literature from yam itself, from other crops, we found that there are possible solutions which can be tried to see if it can help increase pollination efficiency on yam. And that's the first one. We thought on pollen collection and storage. So some activities were done before, but there is need to optimize the protocols which existed, because the other works done before was applying protocols from other crops directly without any optimization activity. We thought also that it should be better to test sites, so to this adequate site selection, so that it can help the crop or crossing blocks in conditions which are conducive for flowering, for food setting, for seed setting. But also there is a importance of using markers. What Dr. Asrat and others discussed that aspect yesterday on sex suite across environment and years. But using markers, it's possible to predict at earlier stages, so that planning hybridization can be much more easier. But also there is a need of profiling genotypes for ploidy status, for cross compatibility and flowering time. In addition to that, we found that there is also need for applying biotechnological tools, such as the embryo rescue, so to break some of those incompatibility barriers among yam species, but also to train technicians to put in place appropriate mating designs, but also to see if there will be a way of rearing insects pollinators, because we found that there is an efficiency also of pollinators involved in yam pollination, because there is also another aspect in yam is that the yam pollen is sticky, so it's sticky to the anther and making the winds pollination impossible. And though the natural yam pollination is relying only on insects, and we found that based on literature that those insects are sometimes inefficient in doing so, which explain the very low success rates when you rely on insects, less than 20% most of the times. That is the background of what I'm working on. For this year now, as Dr. Asrat has introduced, we are trying to test some agronomic and hormonal manipulations to see if it can increase the intensity of flowering, but also to see if we can help also manipulate the ratio between male and females. Those manipulations have been successful on other crops such as cassava, such as potato, and we believe that it could be possible to test. So we are testing them to see if they can give a result on yam too. But also we are assessing the optimum pollination time. Yes, assessing the optimal pollination time. So because we saw that there is that asynchronous flowering between male and female, then we are going to see at the daytime which is the best time for crossing, at the time when the pollen is highly viable and the stigma receptive. But also for this year, we are testing also the pollen storage protocols. We are trying to optimize so that it makes possible the establishment of storage practices. But at the end also, as Dr. Asrat said, we are going also to try to test some markers. So there are markers identified by the yam breeding program, and they are going to see how well they can help predict the flower sex and flowering intensity in yam. It's mostly that one. Thank you for the opportunity.
Okay, thank you, Mundo, for your explanation. We proceed with the remaining plans. So I think it's the last product, flagship product or next generation of varieties. Key traits from either farmers varieties or for gene bank. We are also planning to scan new set of farmer varieties for key agronomy quality traits. Scan new set of gene bank accession or maybe secondary gene pool for key agronomy quality traits. Early bulking, YMBV resistance and YAD resistance. Maybe I think this is those two we expect from the Dr. Lava team to go and scan from gene bank or some other source and then help us with getting resistance source. The seeds for quality seed for scaling trials, we assume that all the partners implement SH to generate quality planting material for breeding lines. We also plan for all partners implement a separate seed flow technique to generate uniform planting materials of test clones for 2020 breeding testing. So for those going to be tested in 2020, then I think you need to use a mini-set technique and to multiply side by side those material to get a quality planting material. I think the one which works for us is mini-set technique, 25 gram, under high plant density you have seen from Dr. Maroya presentation. If you go up to 120,000 plants per hectare, then we can get seed size tubers for the next season planting. Please think about that and try to include all test clones under 2021 trials partly be put under seed multiplication plot so that we just move away from using planting material from tubers coming from trials, so that leave for more room for those tubers to be for food quality and other assessment. And improved population for Diascorea rotundata, we execute all the stage breeding lines advanced from 2020 testing, tuber family, clonal nursery, early generation testing, and also advanced generation testing. I think these are all the stage of the programs in the breeding program, so we keep on moving those materials. The same for Alata. And I think one is very common for all of us is about G by E analysis model. So where we have already distributed the regional variety trials, the RVT, across all the partners. I will try to show it for both Rotundata and Alata, 14 clones. And we included one standard check. So I request for each of the partners to include either their national check or a local check for head-to-head comparison. So it's 4x4 simple lattice, 4 blocks, 2 replication, and a single row of 10 plants, 250 grams set weight, and spacing is 1 by 1. I think there is SOP already we distributed, so hope we will follow that SOP. And then we conduct this one to 2 seasons. Season 1 is 2021. And then we also move the same set of material in season 2 to at least so that we can see the repeatability, the year interaction and location, and then we'll come up as a genetic correlation among our testing location in West Africa. So I think we have a big number. We multiplied seed last year and we are distributing the seed. So in Nigeria for Rotundata, we are trying to include 6 locations, and Benin 3 location, Ghana, SARI 3 location, Ghana, CRI 3 location, Cote d'Ivoire 3 location. Hope they are received, in the process of receiving their material. It's the same for other countries, but since we have more of Alata, we can go for 9 locations in Nigeria, and I added for Cote d'Ivoire 4 locations. Hope they will execute all those. And please try to consider this. We mentioned for local check, the design has been done. And then you use your own standard check for additional 1 check, and don't forget that one. And then for the Nigeria, we try to continue with the MLT 1 and MLT. That one has been started in 2019, I mean, for both to get the requirement of the national variety. At least in 1 location, it has to be tested for two years minimum. So functional regional coordinate threat observation network. We are just proposing for the demand creation trials using recently released varieties. And so I think this is the list for Nigeria. We are planning to conduct, I think, those under over 200 farmers, but I expect also from Jude, he has also multiplied, and maybe he can also put additional 100 so that at least we have over 300 farmers get exposed to our materials. So when we are conducting, we assume, I think I should go to like this one. So like this is the model we are going to use. 1 test variety, and then we ask farmers to use his or her own check. So when we are distributing in 1 village different genotypes, this is farmer 1, this is farmer 2, and so that at least all of the material we are testing be represented in that particular location. And thank you very much. Maybe I think for a small discussion, I took all the time, only 1 minute left. Yeah, I think they talked to Prof. Oh, okay.
Thank you very much, Asrat, for that detailed work plan for 2021. I think now we are all clear as to what we are expected to do in 2021. There was this issue of the trials not being uniform across all the partners. That was, I think, a year or 2 years ago. But now, with this explicit presentation by Asrat, I think every one of us know what is expected of us in 2021. I hope you will share this presentation with us so that it will serve as a guide for all the partners. The floor is open now for discussion or maybe clarity on what Dr. Asrat have just presented. So we still have about 6 to 10 minutes for partners to comment or maybe ask for clarity on the presentation by Dr. Asrat. Yes, the floor is open for discussion.
How is the transfer of the seed is going to be done, Asrat? I mean, is it okay to send mini sets to the partners?
The transfer of seeds, we got all the requirements for the import and phytosanitary, everything, and then
good.
Yeah, so
it's good that you can try. I imagine the requirements they had to be coming from index plant or things like that.
Yes, there are some. I think this phytosanitary organization. They have the receiving countries. They are also listed, and based on that requirement, we got the certificate, and everything I do treated, processed by our the plant health unit, and then we packaged it very well for each of the partners.
Okay.
Yeah, carrying all the documents, I think, and that's how it was. It was even inspected visually by phytosanitary I think personnel at different stage of seed multiplication.
Thank you.
Yes, thank you, Asrat, for your presentation and for indicating that the slide will be passed across to partners. I would want you to indicate on each of the outcomes and milestones, the partners that are responsible. Because when you talk about partners, especially in my own case, the template I received earlier on did not include all the activities for Ebonyi State University, having in mind the resources available to us. So I would want you specifically to identify the partners for each of the activities so that I will know the one we are involved in. Okay.
Thank you very much, Professor Oselebe. Remember that every year we have what we call contract agreement between the partners and IIT on this project. I used to draft the contract agreement, and all the activities you are expected to do are usually in that agreement for each of the partners. So I think immediately after this, our annual meeting, and with discussion with Asrat, I will be rolling out the second year contract agreement, which will now specify what individual partners is expected to do in 2021. Thank you. Okay, we also have the activity tracker that is being used to track the activities of individual partners on this project. So it's not a problem. I will draft the contract and it will be explicit, and you'll know exactly what you're expected to do. And we even follow up. We will follow up with the activity tracker to see whether you are making progress or not. So thank you. Okay, the floor is still open for more comments and discussion.
If I may, Asrat, again, how were the locations selected for the uniform trials across the region? Do they represent, I mean, in each country, the major growing regions, I imagine?
Okay, I think I also need, because I left that one open to the national partners, we gave maximum minimum 3 because of depending on the seed availability, and in order to capture more environment, and instead of going for 3 replication, we made it 2, and then we increased the set size from 150 to 250, so that at least we can get more vigorous plant to improve our data quality. So for Nigeria, we try to represent the environment. The 6 for Rotundata, for instance, representing some from the forest, from derived the transition zone and up to Abuja. And from, I think in Jude also will handle 2 of those, one in Umudike and one in Iboni. Yeah, so try to represent at least that's the environment. And for the rest, they have already, because we are working on the YAM based the trial design, so they suggested their location, and I would like for each of the partners to how they have already selected those their location, and we hope that they will not put within close proximity and represent strategically their production environment. So maybe it's good if we hear from the partners.
I think it's a great opportunity not only to study the adaptation of the genotypes, but to look at the how good the sites are. Okay, and
yeah, that's how I think.
That's a great.
What we do is we develop the SOP, the detailed SOP, what they are going to capture in addition to the genotype performance, the weather, the soil, and then the GPS, the geographic reference point, and all the information. I made clear SOP and hope they will follow, but we're also discussing with them on the trial design, and we also specify the trait they are going to look at a common set of traits in addition to what they want to maybe look for their own purpose. So maybe if we have like from Benny, if you, Professor Dancy, if you want to comment how you selected your trials, or okay, time, yeah. Patrick is saying time. So hope the
that's okay. That's okay. I don't need to have the details, but just wanted to make sure that
okay, so yeah,
we take advantage of such a big effort that you put it into this uniform trial to get the maximum information, not only about the genotypes, but also about the sites.
Yes, and we also genotype those materials, so we have all the information. Yeah.
Okay, thank you.
Thank you, Carlos and Asrat, for that. We are really running out of time. We have 3 more presentations this morning. And there will be a presentation from the IBRC in Japan, Dr. Terauchi, and also a presentation from our partner in CIRAD and our partners in BTI in the US. So I will be, yeah, corrected, yeah. There was a mistake there, I've already corrected it. So we'll be waiting for Dr. Terauchi to be online and present his progress reports for the past year. Dr. Terauchi, are you online?
Yes. Okay. Thank you very much, Patrick. Can you see the slide?
Oh, yes. Yes, I can see the slide. Can I put it
okay.
in?
Can I start?
Yes, go ahead. You can start.
Okay. Thank you very much, Patrick, and thank you for all the people for supporting our effort to elucidate and understand the genetic diversity of YAM. So on behalf of IBRC, I would like to share our latest development of research. And today, Motoki Shimizu, Satoshi Natsume, Akira Abe, Aoi Kudo, and Yu Sugihara on board are also, attending. So if you have specific questions, please address to these people. We have 5 assignments given from IITA, AfricaYam, and I go, each of them in the next slide. The first one is identifying QTL using mapping population and the Jivas analysis. And this is one of the example of MP2 mapping population. And we addressed the pigmentation of purple color in the tuber. And among 156 F1 progeny, we scored the phenotype. And after comparing with genotype, we identified very strong QTL on chromosome 4. And maybe, and we further studied the detail of the genes located in this position. And one of the candidate gene is DRTNTG 13222. And so maybe this is a candidate gene. And using a SNP marker located on that gene, we could divide the population into two with the two genotype on the right side. And there is a clear difference of the color between these two SNP types. Another example is the stem number. And this is a Jivas, not the mapping population, but we found several strong kind of load score after Jivas. And one association on chromosome, I think 5, indicated by arrow. So we further studied the genes located here, and we found two candidate genes. One is cellular synthase, and second is Bryo-like brassica steroid, a kind of biosynthesis gene. And strikingly, the SNP located in this gene lesion shows very clear effect on stem number. So this can be used for the marker to predict the stem number. Okay, I switch to the the elucidation of molecular mechanism of sex of guinea yam. And so as we reported in 2017, guinea yam sex determination is ZW female heterogametic genotype. This is very contrasting to the majority of Dioscorea, which shows XY male heterogametic sex determination. And we studied the genome composition and compared the male versus the female. And this is the on the left bottom, you can see red color is female specific genomic region, and blue color is the male genomic region. And we found around 150 kb region which are specific to female, and we synthesized PCR primer SP16 and used this marker to genotype the parent, male parent and the female parent, and the progeny derived from these these crosses. And you see SP16 is specifically amplified from female, but not from male. And using this marker, we validated the prediction of sex, and in two different families. So the absence of SP16 PCR amplification clearly indicates the male. But on the other hand, the amplification of SP16 does not predict perfectly the sex. As you see here, some shows the female phenotype, some male, and some monoecious. And so this year we added Oxford nanopore sequencing, and we further elaborated on the reconstruction of the W region, as you see in the previous slide. And now we know that the region encompasses around 1 mega base region. And as you see here, this is the association of sex with that region, and there is a high score around this 1 mega region, and this is the depth of the sequence read to the reference genome, and this W region is covered by about half of the depth of the other region of the chromosome. So again, we confirm that the sex determination system in guinea yam is ZW, and female has female specific W region. We further included RNA-seq data to identify the genes which are located in this W region specific to female. And we found 135 genes are located in this W region. Among them, 15 genes are expressed in female flower. And most notably, one gene named DRNTG 3780, which is specifically expressed in female flower in early stage, but not expressed in other stage of female as well as in male flower. So we suspect this gene might be the causative gene in determining the femaleness of the plant. But still we have to go further. And this 3870 gene is a partially predicted gene and similar to proline-rich protein 36-like. And there is a report that this protein might be involved in microRNA biogenesis. So we have to further verify the function of this gene. And we also need to do the microRNA sequencing in this season. So what we are now thinking is that maybe ZZ type or genotype result in the maleness, and the ZW type female has the W region and a gene product, either protein or some RNA expressed from W region, and which suppress the femaleness, maleness, resulting in the female. And if this suppression is not complete, it results in maleness. And if this kind of chimerism occur in one individual, then a monoecious will be resulted. So we still have to go further, but we are getting close to the understanding of sex determination of Guinea yam. Okay, next switch to the pangenome of Guinea yam and its wild relatives. As you can see here, there are 7 to 8 wild species that are closely related to Guinea yam and distributed in West Africa. As you see here, this is Togoensis and Abyssinica, and this is Minutiflora and Lipletiana. This is Prahensilis and Sumilasifolia. They are located, sympatrically, and they may contribute to the pollen. And the underground part of these wild species are very diverse: Minutiflora, Prahensilis, Abyssinica. So they are, very morphologically diverse. And personally, I have been working on the wild species 30 years ago in IITA, and these are distribution of 8 different wild species in Nigeria. And at that time, 30 years ago, I did the chloroplast DNA diversity analysis. And so we are quite confident that these wild species contributed the genome to Guinea yam. So 2 years back, there is a very good report from the Montpellier group, and this was published in Science Advances, and they studied the pangenome of Guinea yam, including the wild species, and they addressed 80 Guinea yam, as well as 29 Abyssinica wild species, and 21 Western Prahensilis, and 18 Cameroonian Prahensilis materials. And they studied the whole genome DNA diversity. And they concluded, so these wild species contributed, or these wild species are the progenitor of Guinea yam. So this is fantastic report. But as you see here, this is a PCA analysis of the report in figure 2. So this is rotundata Guinea yam indicated by red color, and Western Prahensilis and Abyssinica is indicated by this blue and green color, and Cameroonian Prahensilis in this light blue color. So in the PCA axis 1, these 3 groups, Western Prahensilis, rotundata, Abyssinica, come together. So they are very close. So this is one message. But in the same report, they provided a phylogenetic tree showing rotundata is most closely related to Western Prahensilis and next to Cameroonian Prahensilis and very distant from Abyssinica. So these 2 data, one in PCA and one in phylogenetic analysis, does not are not consistent to each other. So we found that maybe something is still missing from the analysis. So we included 308 Rotundata accession whole genome sequencing, and together with the genome sequence of wild species reported in this Science Advance paper, we reanalyzed the genetic diversity and published the result in PNAS last year. So this is a summary of the diversity of 308 Guinea yam accession. You can see this theta pi. So this indicates the genetic diversity of Guinea yam. So you can think like if you take two chromosomes from Guinea yam cultivar and compare their sequence, there is on average 1.5 SNP per 1,000 base pair. So this is not so high, but very similar to the genetic diversity of rice, for instance. And after the structure analysis of this Guinea yam cultivars, we found the cluster 1 through cluster 5, and the cluster 1 seems to be the hybrid of Guinea yam diploid and Dioscorea togoensis wild species. And we focused on the cluster 2 to 5, and we made a phylogenetic tree using the SNP data. And as you can see here on the bottom, so these the groups indicated by red or pink color, these stand for the Guinea yam cultivar. And this yellow one is Abyssinica wild species, and the blue one is Prahensilis from Western Africa, and the green one is the Cameroonian Prahensilis. So our data suggest Guinea yam cultivar is most closely related to Abyssinica wild species. So this is not consistent with the report in Science Advance. So we further analyzed the past demographic history of Guinea yam and the wild species using the DADI kind of bioinformatics analysis. And as you see here, this red, this is a likelihood value. And this P stands for the Prahensilis from West Africa, C stands for Cameroonian Prahensilis, and A stands for Abyssinica. And among the three wild species, so this red color indicated, the red color is reported by the Science Advance paper, and the likelihood is not extremely high. On the other hand, if you group this Abyssinica together with Western Prahensilis, the likelihood becomes higher. So we think this, the scenario that Abyssinica and the West Africa Prahensilis is closer is more strongly supported. And to cutting the long story short, we found that Abyssinica and Prahensilis both contributed to the genome to Rotundata, as indicated by R here, have the highest the likelihood. So we infer that the Guinea yam cultivars are derived from the hybrid between Abyssinica wild species and Prahensilis wild species. So this is the scenario supported by our analysis, that Rotundata located here is derived from Abyssinica and Prahensilis. And so you can refer this paper for the detail, but so final kind of scenario and the model, which is supported by statistics, is shown here. And first, there is a common progenitor of Abyssinica and Prahensilis here, and it was diverged to Abyssinica and Prahensilis. And after quite a long time, they crossed to make the hybrid, which is the progenitor of Guinea yam Rotundata. And what I want to draw your attention is that this NA, the number indicated in the parenthesis, this is the effective population size, predicted size. And Abyssinica and the Prahensilis have a substantial amount of genetic diversity. But by contrast, the Rotundata effective size is very small. So what I want to say to you is that there is a huge untapped genetic diversity in wild species, which can be deployed for the improvement of Rotundata for the future breeding. And this is the kind of summary of our hypothesis. Protein data we are growing now is derived from the hybrid between Abyssinica and Praensis. And Abyssinica seems to be the maternal parent. And so, okay, I skip this. So in the past, we analyzed the Guinea yam crop, the cultivar together with the wild species, and it was revealed that the Guinea yam has been derived from hybrid. And we can look into the future that we should make use of this diversity of Abyssinica and Praensis to find allele mining and to utilize this untapped genetic diversity to improvement of yam in the future. And in this regard, I want to stress, yesterday I heard that Dr. Barada explained about the genetic material, genetic resource, in vitro conservation. And I think these wild species are very important targets to think about in vitro preservation, because the habitat, natural habitat of Praensis, this is rainforest, they are now deforested, and the habitat are now shrinking. So we have to think about preservation in vitro. Abyssinica is a savanna species, so you can find anywhere in the savanna of this wild species, so it's not a big problem. But I want to suggest to consider the conservation of Praensis germplasm in vitro in IITA. So this research result has been published, and the next year's plan is the number one. We want to continue QTL analysis on Jivas together with IITA researchers, and the second one about the sex determination system. We want to validate the gene I shared today, and also we want to add more Oxford nanopore sequencing, RNA-seq, and the microRNA sequencing to finally confirm the determinant of the sex of Dioscorea rotundata. And in the end, we want to validate the gene function using CRISPR-Cas9 mediated knockout. And we may have to collaborate with the Dr. Sripada in IITA Nairobi. And the third one, about RNA-seq, we are thinking about continuing RNA-seq of different tissue and under different conditions together with IITA people. And fourth one, about the pangenome, we still have to enlarge our sequencing effort, and we are going to focus on resistance gene NLR using this data of sequencing. And finally, we want to expand our the machine learning genomic prediction pipeline using CNN or decision tree or other methodologies. Okay, thank you very much for your attention. That's the end of my presentation.
Thank you very much, Dr. Terauchi, for that beautiful presentation for the activities of 2020, and looking forward to what you are going to do in 2021. Thank you very much. We will leave the questions to the end of the presentation, because we're already eating up the time of the next presentation. So the next presentation will come from Dr. Connet in Sierra in France. Connet, are you online?
Yes, I'm there. Hello.
Yeah, you have the floor, please.
Okay, let me share the screen.
Yeah.
Okay, can you see the screen?
Yes.
Okay, thank you.
The screen. Go ahead with your presentation.
Okay, thank you. So my name is Denis Cornet, and I'm crop physiologist at CIRAD, and I present a progress report and a short work plan for next period. I will present with the help of Gemma Arnaud, who will speak about QTL research. So CIRAD is working on the two first flagship products to develop and transfer medium and high throughput methods for morphological quality traits. We are also in charge of organizing and attending a joint training on methodologies for sensory evaluation, textural determination, and food quality assessment developed by the RTB Food Project. This is the work of Dominique Dufour. Within the flagship product 2, we work on identifying QTLs and genes controlling key traits regarding competitiveness against weeds. Just a short parenthesis on enhancing human capacity at CIRAD on YAM research. Within the last year, we hired a lot of new staff. One field technician to increase our capacity of phenotyping, one quantitative geneticist, Komivi Dosa. We also start a PhD within the framework of the RTB Foods project, but in direct link with AfricaYam. The PhD is working on determinants of yam quality. This includes genetic determinants, but also environment and practices determinants. So this thesis may bring some answers to the questions raised yesterday. Can we make a link between vegetation indices, leaf analysis, and so on, and tuber quality? We will look at this within the PhD of Ezekiel Ungo. And finally, a postdoc, Anna Zotamota, who is working in genomic. He also joined the team within the RTB Food project. So first, we aim at developing and transferring some medium and high throughput phenotyping methods for quality traits. 3 SOPs were written within the RTB Foods project, and we shared it with NRCRI at first. I think in the next days, Dominique will make them available on the RTB Foods platform for everybody, but I can send private copy and discuss with people interested in. So the SOPs are dealing with color characterization. The first one is dedicated to allow the user to build his own color reference chart. Starting from his pics on his computer, he can extract relevant pixel and build a color chart, which can be helpful, whatever, if we work with image analysis or if we go for visual evaluation, the reference chart is always useful. The second SOP is there to characterize color globally. I said globally because we are working on the whole tuber. We cut it longitudinally, and doing this, we can characterize color even if the tuber surface is heterogeneous. So we can quantify this heterogeneity, radial or longitudinal, and we can extract different color indices characterizing the tuber, whatever it is, white indices, yellow indices, brown indices, and so on. The last of the 3 SOP describe how we can extend the previous methods to make some color over time, some picture over time. So we can, with this method, extract color indices or color differences at different time steps. And we can study color dynamic, browning, and so on. So this is just to illustrate the point. So this is how it works. We take picture at different timestamp, and using image analysis, we can extract color indices and discriminate between genotypes in order to look to G by E or QTL study. We started also to work on tuber shape related traits. So we wrote protocols and we shared it with CIRAD, INRAE, and CNRA technician to start a study. We didn't share it more broadly up to now because it is still at the proof of concept step. So we are trying to relate it shape traits with quality traits like peeling or harvesting time, peeling yield, and so on. So we used last year harvest to make our first observation on peeling time and peeling yield on 2 sites on a diversity panel of 55 genotypes of the Alata. We also gathered some information about harvest time for each of these genotypes. But the number of observation was quite limited because of the COVID impact. Field was hardly accessible during the confinement. And also we had to postpone support mission plan to help to start this kind of observation at CNRA in Ivory Coast. So it is not cancelled, but it is postponed as soon as the sanitary crisis will allow it. To compensate this, we submitted a grant and we get the fund to be able to host a CNRA partner at Montpellier during 2 months in order to enhance capacity on phenotyping. So he will be trained on image analysis and phenotyping methods. And still with CNRA, we made a publication which can be useful to answer some of the questions we discussed yesterday regarding the different threshold of dry matter or starch and so on, which can guide breeders to make some varietal selection. And we come very close to what Asrat said yesterday. So we look at different thresholds which are necessary but not sufficient. Like dry matter, we know that under 25%, yam will never be good for pounded yam, for instance. And still within this publication, we made also some NIRS model for composition and texture. So just a few examples of the result in the publication. The black dots are the moldable genotype, and the gray dots are the not moldable genotype. And we can see that if we want to have a chance to select moldable genotype, we need some certain amount of starch, dry matter, and hardness. This is a necessary condition, but this is not sufficient. You can see that in between this black dot, there is also gray dot. So it means this is necessary, but not sufficient conditions. We built some near-infrared spectroscopic model in order to be able to predict these traits. We had very good results for dry matter, protein, sugar, and starch. And we had very poor results regarding textural traits, hardness, cohesiveness, and springiness. So for these last 3 traits on texture, we go for deep learning algorithm and we go for classification between hard, soft, cohesive, non-cohesive, and so on, and moldable and not moldable. And this is just to illustrate, we were able to build some very accurate model. For moldability, we have an accuracy of more than 80%, and for cohesiveness or springiness, we are up to 95% accuracy. The second point is joint training, RTB Foods and AfricaYam joint training on yam quality characteristic. So this was made by Dominique Dufour. I will not spend too much time on it because Emmanuel already highlighted a lot of our collaborative effort in the RTB Foods. We just list different main results that can help AfricaYam project to go further. So there was 2 studies on quality characteristics for ready-to-eat products, one for boiled yam and one for pounded yam. There's 2 publications associated. There was one SOP for cell wall polysaccharides characterization from yam. And also 2 SOP in preparation for yam textural standard of operation, one for cooking time and one really for texture. So this one is still in active development in Guadeloupe and Montpellier. There was 2 SOP and 1 training on yam sensorial analysis. And finally, a lot of SOP on yam spectral and image measurement. So this is a list, and we can come back to each of these one afterward if people are interested. I will give the floor to Gemma to present this few diapo. Gemma, are you there?
Yes, I'm there. Hello, everybody. We could identify a total of 34 QTLs controlling K tuber quality traits and explaining between 11 and 43% of total phenotypic variance on the two biparental population generated at the beginning of AfricaYam projects. We identify QTL for tuber size, 5 loci on 3 different chromosomes for regularity of tuber shape, also 5 loci. And for tubercular rods, the tubercular rods are the rods on the surface of the tuber. There is a trait difficult, peeling is more difficult when there are roots on the tuber, and also it's inesthetic trait. And we identify 5 loci, 3 different chromosomes. Also for skin texture, it's smooth or rough. We identify 5 QTL, and for flesh color and the oxidative growing of the flesh. 5 different QTLs were identified and 5 different chromosomes. You can see in the right, the 2 QTLs identified for skin on 2 different chromosomes, the 16 and the 15. And you can see the height lot scores. In the left, you can see the distribution of the different traits in the 2 different populations, in green for population A and blue for population B. And high diversity was observed for all the traits, and the distribution is normal for some traits, but for other it's Bayesian. And we observe for some traits, in particular, the tubercular rods and shape regularity, transgressive hybrids with values either the both genitors, in particular in population B. Next, please. And we validated 9 QTL on a diversity panel composed of 28 varieties. That shows that the QTLs identified was not specific of the genetic background of genitors, and it will be possible to use this for selection assisted marker. And QTL were validated for the tuber size, 3 for shape regularity, 2 for skin texture, and 1 for the presence of tubercular roots. And you can see here the box plot of the distribution of phenotypic data in the validation panel according to observed genotypic classes. The first, it's the alleles controlling the ratio length width, and the other 2 are the alleles controlling the shape regularity. And depending on the loci, we observe 2 or 3 genotypic classes in the diversity panel. And for in particular for tubercular roots, it's a locus with dominant effects. We found only 2 classes in the diversity panel, but 3 classes in the population, the biparental population, and we imagine that it's a contra-selection because the homozygote classes containing the allele that increase the growth of the skin has been contrasted because it's negative trait. And next, please. And also another very interesting thing, it's because we found that about half of the QTL were mapped in the chromosome 16. We analyzed in detail this chromosome, and we found a paracentric inversion on male Kabuza of about 280 kb. What is the paracentric inversion? It's a chromosomal rearrangement involving two bricks and the reinsertion of the chromosome segment after a rotation through 180 degrees. So the order of the genes is reversed in the inverted segment. You can see here in pink the inverted segment. And we analyzed pollen fertility of both males by Alexander blue staining, and we found that fertility of Kabuza is lower than second male, about half. And also we found that the ratio of generated embryos for the number of pollination it was lower in population concerning Kabuza, population A, than the population B. In conclusion, it is possible that structural variations are the cause of the low fertility of the Alata species. And it will be interesting, we have sequences of about more than 100 varieties, but with genomic approach will be possible to study this in more in detail. Thank you.
So, thank you, Gemma. So, still in the flagship product 2, we aim at identifying QTL and gene controlling key traits regarding competitiveness against weeds. So we focused on this important breeding objectives because yam is have a very slow development and is planted at very low planting density, less than 1 plant per square meter. So also regarding its architecture, it means that all the resources you look for come from a very thin volume. Whatever it is for water, nutrient, or light, all come from a very thin volume. The rooting system is very shallow. It hardly go deeper than 15 or 30 centimeter maximum. And because of its crawling habit, when it's not staked, it's hardly get higher than 15 centimeters. So this allows for a maximum of competition with weeds. And all the weeding is hand weeding because there is no herbicide authorized, for instance, in Guadeloupe, or it is barely accessible in West Africa. So it means that hand weeding is there for more than 30% of field labor. And if it's not well done, it can decrease yield up to 90%. Some studies just show that at farm level, hand weeding capacity is often the first driver that limits the cropping area. So the farmer chooses the surface regarding at his capability to make the hand weeding necessary. So this is why we focused on it. To study these traits, we've planted the 2 mapping population already used for quality traits, and 1 diversity panel that was planted in 2 pedoclimatically contrasted sites. And we started some observation last year, but this was limited due to COVID impact. Planting was cancelled by 2 months, and some observation on phenology were cancelled because of this delay. And the ground cover dynamics that we plan to look at on all the experimental design, we only looked at the diversity panel. We were not able to look at the departmental mapping population. To compensate this, we used the historical data that we had to continue to improve the script. So the idea is to make an image with a smartphone or whatever the camera of the same portion of soil every week or every 10 days. We developed scripts to be able to recognize automatically the squares that we put, the rectangles that we put on the ridge, to extract the relevant portion of the image And if there is a perspective distortion, we are able to correct it before going further to other analysis. So the next step, when we have extracted the interesting part of the image, is to make some segmentation in order to extract the ground cover value in percentage. And using nonlinear modeling, we can follow the ground cover dynamic and extract biologically sounded parameters like early growth vigor, like time necessary to reach half of the ground cover, or maximum ground cover, or even the speed of the senescence part of the crop. So there is still some improvement to bring to this script and to gather all the script in one to make it available, but this is already well advanced. For period 2, we'll continue to harvest and plant the parental mapping population and the diversity panel. Gemma will finalize publication on quality traits from AfricaYam phase 1. We'll continue the proof of concept on tuber shape related traits. We will make the training of CNR staff on phenotyping using image analysis, thanks to support mission and the hosting of a visiting scientist. Dominique will organize the RTB Foods AfricaYam II joint training. And we'll continue to phenotype for color, oxidation, peeling yield and time, harvest time, ground cover dynamic, and we will initiate Jivas on diversity panel for ground cover dynamic. We'll write an SOP on ground cover dynamic using image analysis in order to be able to transfer it to partners. And we continue the method development on early stage vigor and crop phenology traits using image analysis. Thank you.
Thank you very much, Aaron and Dennis. Can you just stop sharing your slides?
Yes, let me stop it. That's
okay.
it.
So thank you very much for that presentation, both of you. We're moving to the next presentation, which is from Lukas from DTI. Lukas, are you online? Hello?
Yeah, I'm here. Yeah, I'm ready.
Okay. Yeah, please, you have the floor. Please just go ahead. We don't have your presentation yet. Please send it to us after your presentation.
Will do, yeah.
Thanks. You have the floor.
Okay, I'll share my screen. Can everybody see the screen?
Yes. Yes, we can see your screen now. Yeah, it's fine.
Okay.
Go ahead, yeah.
So I'll give a brief update about progress at the Boyce-Thompson Institute, and I'd like to thank all the members of the lab, and of course also the entire AfricaYam team. A database is of course a community effort, and everybody contributes to that. And so thank you. If you now I have to figure out how to, yeah, okay, it works. So the goal is, as you know from previous presentations, that we want to create a digital ecosystem for YAM in YAMBase, and basically all the processes are digital in the system. You can create field layouts and evaluate phenotyping, and you can collect samples, and that's usually done with barcodes on the field and tablets, and then you can do your analysis, and you can perform the crosses and make the selections, all with a connection to the database, so that all the information is always up to date in the database in real time. And so that's the idea, and I think we're almost there. We have almost, all the different parts are together. And so, of course, the database needs to be used to be useful. If you have the best system and nobody uses it, that's not useful. And so this is the current database stats. So we have almost 500 trials in the database, and that's 143 trials more since March 2020. And I find it actually quite amazing. It's been a very, very difficult year, as we all know, and still, I think a lot of data has been submitted to the database. You can also see here with the stock types, there are new accessions. So these are mostly products, and that's an assumption at this point, from new crosses. There are 13,000 new plots in the database and almost 6,000 new plant entries. And in terms of traits, there are almost 200,000 new trait measurements in the database. So things are, being added, and the database is being used. I'm sure, we can still increase that and improve that, but I think we can say that the database is active and running. So I would just want to talk about some recent and current work that we are doing. And I think Asrat has been mentioning the stock catalog feature that we want to implement, and we're currently implementing, and you see some early designs here of the user interface. And so what we want to do with the catalog is showcase the best clones, and we have a separate search for that. That's very simple. It also will then lead to a separate detail page that doesn't contain all the data that a normal clone page contains, but will be a much smaller page and will highlight the clone with an image that you can select, and so on. And another feature that's related to this is that you can then order a clone. And so the database will know which clones are available from which centers, and then there will be the ability to have a little shopping cart, and you can send the order to the clone centers, and they can log in and they can see their orders, and they process their orders and update the statuses and so forth. In the end, of course, send the clones and finish the transaction. Another important thing that we have added is product profiles. I think product profiles are extremely important in breeding, obviously, because it really presents the breeding targets and the program goals. And I think that's a really important connection between the data and the goals. And so in BreedBase or YamBase, you can now specify the trait values that are in product profiles that are the breeding targets. You can do that either by an actual trait value or a reference accession. So every breeding program can do that. They can define one or more product profiles. They can have several product profiles that maybe define different market segments and so on. And so that's what it looks like when you go to the breeding program section. You have a new section called product profiles. You can upload a new product profile. Currently, you have to create the product profile data in an Excel file, and then it will be displayed here. And you can search them and so on. And then you can look at the details of the product profile. What we would like to do is that you can search the product profile in the database and match, the accessions that meet or exceed the product profile specification, but that is not implemented yet. So that's what we are working on. Another cool tool I think that we have added is a field Geo Viewer and editor. And it's essentially a tool that's based on the BRAPI standard. And we have co-developed this with Noel Soldvila and Mariano Crimi. And what it does, it essentially overlays field map information on satellite imagery. And that's a way where you can actually get fairly precise plot geodata into the database, which is important for many applications. So this is what it looks like. When you go to the trial detail page, there is a button. And if you click it, this page will come up. It will because we have the location information of a trial in the database, it will go to that approximate location, and then you can import the field plots, and you can adjust the field plots to match exactly your field in the using the satellite imagery as a reference. And then you can store the coordinates in the database. And it's a really cool and interactive tool. It's very nice. So during the last couple of years, we've actually added a lot of tools. We have added things like a heritability tool, a stability tool with AMI plots. We have, of course, the mixed model tool that we added essentially because Azrat was asked us to do it, and so we added it, and many other tools, and this really has led us to think more about raw data and analyzed data and the relation between the two, and so of course if you have a lot of tools, you need a way to query the data and specify the input data for the tool. And we have, as most of you know, we have this universal query engine on the database that we call the wizard. And you can make selections, and essentially the wizard represents you with these 4 columns that represent the dimensions of your data. For example, here I have selected years as a dimension, and you can select a couple of years, and then you can go on and select other dimensions. For example, here are locations, and then trials, and so forth. And what you can do then is you can use these data selections to immediately download related data, such as genotype data or phenotypic data. But you can also save this search parameters as what we call a data set. And then this data set can be input into other tools. Then, of course, you would like to, once you have run your tool, you would like to store your analysis, right? And I think previously what happened is that we had tools and you got a result, and that was the end of it. You had to somehow copy and paste the results, or use them in some other way. But now you can store the results directly in the database. What we call this feature analysis. So we can store all the analysis metadata in the database. So we have seen that the data set from the wizard actually kind of represents the data an analysis is operated on. We can store the protocols, and we can store even the models that a specific protocol is based on, and we can store the results in the database. And so now I think that's very interesting, because we can also chain analysis in the database. So we have maybe raw data in the database, and you can analyze this data in some way, maybe a field spatial correction, which is a tool we're working on as well. You can maybe do a mixed model analysis, you can do predictions, and then you can feed the analyzed data into your breeding decision algorithms. So I think that's a very interesting way to think about data. And so we have a number of tools that already implement storing of the analyses, and that includes, that includes the nearest calibration that I will talk about in a minute. We have a drone image analysis tool that uses this, the field spatial correction tool, and the mixed model tool, and also the SolGS genomic prediction tool uses that. And this is what it looks like. You can go to The database here is a screenshot from cassava base, but it's the same in yam base. The first item in the analysis menu is now a analysis and models item, and when you click on it, you come to this page and you see all the analysis and models that you have in the database. You can click on the individual analysis, and you go to an analysis detail page where you can look at all the data. It prints you histograms, and you can use other tools based on this analysis data, on this analyzed data. I want to briefly talk about genotyping information because we have made a lot of progress as well in managing genotyping information. And we have published actually a paper, the high density genotype storage for plant breeding in shadow schema of breed base. And we have published this about 6 months ago, and since then we have already made other improvements that are very technical in detail. We also added the ability to store SSR data, and SSR data is quite a bit different from the SNP-based genotyping data that comes out of DART-seq or GBS, because it has a lot of metadata such as primers and so on. And so now you can upload all this. So you can, in the genotype upload, you select SSR, and there's a specific file format, and you can upload the metadata such as the product sizes that are possible, and the forward primers, reverse primer, annealing temperature, and so forth, sequence motif. And then you can upload the data for individual lines and specify which band sizes are actually visible with these specific lines. We've also improved how you can submit genotyping plates. Now we have implemented a submission protocol that is based on the BRACI 2.0 specification, which is supported by DART and Cornell currently, and I think Intertech, as Nhua has told me recently, will support this by the end of the year as well. And so there's a specific workflow in the database. You can generate samples and plates using an app called the Coordinate app, which is from the same people around Jesse Poland in Kansas State University as the Fieldbook app. And you can go and collect these data. It works with the barcodes that you already should have in the field, and you can select the samples, and you can then upload these data to the database. It will generate the plate representation in the database, and you can then submit that plate through the database directly to a compatible provider. This is just the whole process. I'm not going through this in detail, but it's an online process. You submit it to the database, and then when you go back to that plate page, you will actually see the status. So every time you open that page of that plate, it will go and query the status of that plate at the provider and give you the up-to-date information if it's in progress or if it's completed and so forth. If it's completed, of course, you get the download link, and you can download the raw data and process it and upload it to the database. This is a wrong slide. For genomic prediction, we have created a tool called SolGS for cassava base, and that is also a tool that we want to make available in yam base. So currently, we have heard a lot in the past couple of days about genotyping that people did, but unfortunately, I find that not a lot of this genotyping data has made it into yam base. So this is really something we have to work on. I know that some of these data, of course, are still subject to publication. And so clearly, we don't want to store it before it is published. But I always recommend to use the plate submission system in YamBase so that we can easily connect the genotyping data to the accession data in the database, because that's what you can do when you use that system. Otherwise, it can be very hard to actually match up what you genotyped with accessions in the database, because you may have used different names and so on, the usual database problems. And so it's important to always use this digital ecosystem to prevent these problems and headaches later on in uploading the data. We've also been working a lot on crossing data collection, and we support two crossing apps. One is YamCross that has been adapted from Btract, which is the main supported tool. And we have also, for other databases mainly, we're also supporting the Intercross app. And crossing has become really a very well-defined and a very powerful system in breedbase. And I think maybe at a future workshop we should talk more about it, but I just want to mention it here that this is an area where we have made a lot of progress. This is just an upload of the Intercross file so that you see that that's implemented and works. But I think YAM is really standardized on YamCross. So one important thing is a new focus on quality data, which of course is very important. And here we work a lot with RTB Foods and the crop ontology programs to add the required new traits to really describe quality data, as well as new ontologies, because what you need to do, of course, is you need to describe the food products in more detail. It's now not only a certain accession that's growing in some plot in the field, it's now has undergone some treatment like cooking or boiling or roasting. And this needs to be represented, of course, as well in the database. And we're working a lot with Elisabeth Arnaud and Marie-Angelique Laporte, as well as Thierry Tan and Karima Megar from CIRA to add these traits. So this has been a very nice collaboration. And so one example is of an ontology you have to add is the treatment ontology. And this is, of course, quite specific also to each crop and so on. And so you know, you wanna define that, and we are currently doing this. Another thing we have added is what we call the high dimensionality phenotypes. And one important representative of that is the NRCRI data. And we have heard a lot yesterday about NRCRI and how it is revolutionizing, you know, phenotyping in many crops. But it's a very complex data type, you know, because you're really collecting spectral data. And with the spectral data, you have to generate a calibration. And from that, you can then deduce specific traits. And so all this needs to be stored. And YamBase can now actually store the spectral data. It can perform the calibration in the database. And it stores then the raw data and the predicted trait data in the database. And I just want to mention that this is really based a lot on the work of Jenna Hirschberger from the Gore Lab at Cornell. And Nick and Chris have worked with her to integrate her tools into cassava base and related databases. There are actually a lot of other high dimensional phenotypes we have discovered since then. So for example, we can measure gene expression data and we can store that now in the database, or protein or metabolomics. Another data type that cassava is now testing is ground penetrating radar. And that might be also something obviously that's interesting for YAM. I'm not sure when, you know, if there's any plan to apply that to YAM, but I think we're working on a system that can deal with it and can store that data as well. So for 2021, I think what we really need to do is we need more training and workshops, and maybe I feel a little bit more interactivity between the breeding programs and the Yambase. There are certain programs we interact with a lot, and certain programs where we don't interact that much. And I think people have said we need from the breeding programs, as I heard yesterday, is that we need more interaction between Yambase and the programs, and that's completely true. And I think we should probably have some formal workshops because there are so many new features like product profiles. There are not a lot of new features in the crossing tools. So I think it would be really worth holding some workshops. But it also, if you have any kind of problem with the database, don't be shy to contact us. We are here for that. We will try to resolve any problem. And breeding data is very complex. It's important that you get the support that you need, and we want to provide that. We work on another set of tools in the database. I've mentioned the field spatial correction tool, which I'm kind of excited about. We also found that we probably need sensory panel trials in the database. This will be a special trial type in a way. So Yambase will think about sensory panels as trials. And that's something we are working on. I've mentioned the clone catalog and ordering system. That's I think that's quite a large piece, and it's already well progressed. So I think we can have when we'll have organize a workshop, we can talk about this as well. And I think we're also very excited about high dimensionality phenotypic data that I mentioned, and in particular, the ground penetrating radar data might be very useful for many root crops. I would just like to thank the people in my lab. This is a somewhat older photograph, but especially I will also thank to all the people of AfricaYam who make this, who make this database what it is. So thank you.
Thank you very much, Lukas, for that detailed presentation. We will reserve all the comments and questions to the general discussion immediately after lunch. We'll be moving on to the next item on the agenda, which is the general discussion and meeting with TAC. At this, I would like to announce that the next 30 minutes will be offline for the majority of the participants. So it's only the TAC members that will be online for the next 30 minutes. You will rejoin at exactly 12:30 for a general discussion for the TAC members to the team. So the next 30 minutes, majority of us will be put offline, and we'll be having a kind of a meeting with the TAC members. Thank you very much. The TAC members, I believe they are already online, and they'll be I'll be having a discussion on their general impression about the project and the advice they can give to the project management. After that, we'll ask you to rejoin. We'll open it again. You will all rejoin, and the TAC members will speak to all of us. Thank you very much. I was told that we will be in the waiting room. After our meeting, we will readmit you back again into the meeting. Thank you very much. I think you can, the IT, yeah, you can just leave the TAC members online. Thank you. Don't forget, in 30 minutes, you are rejoining us, please. Yeah.
Patrick, this is Carlos. Can you hear me?
Yes, Carlos. Yes, I can hear you.
I'm transitioning off.
Hello?
Hello?
Yes, go ahead, Carlos.
No, I'm transitioning to my office. I'm driving to my office, so I'll be on the phone, but I'll be off camera, okay? But I'm here.
Okay, okay, that's fine.
Hello, Davis. Dave.
Hi there.
Hello.
Good to see you. Hi, Noel.
Hello.
I'm not sure, is Eric with us?
Yes, sir. I'm Noel from Benin.
No, I'm wondering if Eric's with us as well. Yep.
Yes.
He shows on the screen.
Yeah.
Yes, hello, David. I just joined in actually. Good morning from Accra.
Okay. I mean, I think we've got quite a difficult task to actually give a sort of major feedback at this stage because, I mean, I think ideally we would have had an ability to sit down and talk as a group more, you know, several times before this. So I would suggest that, not sure if we can find a way to stop that feedback. I would suggest that we have a brief general discussion now, try and identify some points that we can relay, but I think they'll have to be relatively high level generic points. But Patrick, I think it would be very sensible to try and arrange a proper meeting of this group beyond this current session.
Mm-hmm.
Patrick, if you could mute the camera room, the IATA camera feed, I think that's where the feedback's coming from.
Well then,
The feedback.
I think I've got to start off with saying that that clearly there's going to be a need to update your working plans in order to realistically account for what hasn't happened because of COVID. But we'd also say that we're incredibly impressed with how the AfricaYam team as a whole has coped with the issues involved. I know.
Sorry,
example,
David, could you share your slide?
I was just thinking I was just going to read it out rather than sharing it. I think because I
Okay,
may add
sorry.
a little bit as I go along, provided people are happy with that. I know what the reality is with the laboratories in the UK, many of which have almost completely stopped working, apart from really emergency work. And I'm really impressed with how the team in AfricaYam has coped. We do know that the existing work plan is extremely ambitious in terms of the amount of time and resources that are allocated. And so I think people need to be aware of what the actual changes that they might need to put in place so that you can make realistic use of the resources you have available within the timescales that are going to be available to you.
The second item that I would like to raise is that you're not that long into a second round of Gates Foundation funding, but already you need to think about beyond that. So you need to start to at least have some thoughts as to what will happen next beyond the end of the current program. And this needs to take account of the reality of the resources that you have available to you. It also needs to think about how you might want to lobby for more resources, both locally and internationally. Clearly also the CGIAR is going through a transition. And again, I think it's important to identify whether there are things that are going to change around you in terms of the CG system and what it really reflects in terms of projects like the one that we're involved in today. One of the key things that is also going to be is to get the balance right between the support that can come from IITA and the degree of independence that the NARs are going to be able to generate based on resources that have come from this project. There's been, vehicles made available to you. There's physical resources in terms of laboratories and yam barns. But you're going to really have to think about how you're going to be able to resource this the work you do longer term. I was pleased to see from the work that Asrat talked about today that you're putting in place quality control sampling with markers in terms of your plant material at various stages in the breeding program and the gene bank. I think this is incredibly important. I work with a range of tropical crops, many of which are clonally propagated. And every time we've actually gone into either breeding material or gene bank resources, we found that there's a really quite frightening degree of mix-up in terms of samples. This is just a fact of life. But the great thing about being able to do quality control sampling is that you can identify where the weak points are in your processes. You can identify the particular transitions where you have to take a special care, and you can quantify what the degree of problems that you actually have. So I'm really very, very pleased to see this. And thinking about quality control at every stage of your operation is really important. Carlos tells me that NextGen Cassava has implemented the concept of quality champions. And this is something that may be really important for you folks to think about. As a group, we've been really pleased to see the new quality work coming on board in terms of this project. This is something that we advocated in the phase one of the project, and so the concept of both project profiles and looking at quality in yam, we believe is really important. I'm really pleased to see that being done. It will be really interesting to see how well it scales to a larger number of samples, because that can be really important in terms of supporting the breeding programs. We are pleased to see that there are a number of new partnerships that have developed, but also we believe that there's considerable opportunity to broaden the partnerships that you have. This is partially about advertising what you're doing, but it's also about trying to identify skill sets in laboratories around the world which are relevant to the work that you're doing. And this is important in terms of bringing new capacity to you in terms of skills, equipment, but it's also an important way of leveraging new funding that can come into YAM. A clear opportunity is the whole issue around flowering and seed production. This is one thing where it's really important for what you're doing, and the more groups who have skills and experience in this area that you can bring into your project, the better. The one thing that we would really like to see also is the generation of outreach material that explains your project goals, the research you're doing, the outcomes, so that both your partners and collaborators can use that to educate colleagues and decision makers about the relevance of the work that you're doing and the importance of YAM. Outreach is incredibly important. And certainly the science I'm involved in the UK and Europe, there's been a dramatic change, I think, in the last 5 or 6 years in terms of scientists realizing that until people know about their work, it's not really done. And so outreach is an incredibly important part of what you do. So again, we would really like to advocate that you think quite carefully about what you do in terms of outreach and how important it is in terms of your planning framework. That's all I want to say at this stage. I'm not sure if any of the other partners on the Technical Advisory Committee would like to comment, but I just want to finish what I say by saying that I'm really, really impressed by what you've done under difficult circumstances. And we hope we're going to carry on with success.
Yeah, thank you, David. I will echo David's words, and although we had a brief interaction with him last night, I think we agree on several of the points that he made. One thing I wanted to stress is that we would like to have a more fluent participation, and not only maybe not so much because of what we can contribute to the project, but we can learn from you guys and try to find or help you find solutions for your issues or for your tackling opportunities. So we raised this with Patrick. We do want to have a more frequent interaction, not only with the project managers, but management, but with the project as a whole. So I hope we can do that through the year. That's all I have to say, David.
Would any of the other members of the TAC like to make some comment? If not, then I think we can finish off there, Patrick.
Thank you very much for your comments, your suggestions, and we have taken note of all those course corrections, especially in the area of advocacy that we have mentioned. We try to make it more visible by working very closely with the Federal Ministry of Agriculture and Rural Development, not only in Nigeria. We'll also try and work with relevant people in Ghana, Benin Republic, and Cote d'Ivoire. Thank you very much for all those comments. Thanks, David, for that. I think we have gotten very valuable suggestions from our TAC members. I will be expecting David to send me those slides so that I can document them and also remind me of what to do. We will schedule the kind of meeting with the TAC and the major stakeholders in this project. At least 2 meetings will be scheduled for this year, so that we can keep you abreast of what is happening and get the relevant technical advice from you. Thank you very much. On this note, if there is any other comments from those that are here today or those that are online, we still have about 10 minutes. If there's any other comment from anyone, there's still a chance to make comments. Otherwise, we'll be meeting exactly 1 hour from now after lunch. I didn't see any hands off. So thank you very much, Carlos. Thank you very much, David. Noel, thank you. You are attending the meeting for the first time. Thank you very much.
Thank you very much. Thank you.
Thanks.
Thank you very much. Yes.
Is Jim still online? Thank you very much. We'll be expecting you to give us your feedback after lunch, exactly 3:30. So thank you very much. I think I can ask everybody now to go for lunch. We'll be meeting exactly 1 hour from now. Thank you. So we'll come back at the 4:00.
You are welcome for the last section of the AfricaYam meeting annual report, and then planning progress also. So this afternoon we'll be trying to have the comment from the, we get to the general discussion, and then also we have a reflection and key action point from this senior program officer from Bill and Melinda Gates Foundation. And the closing remark will be done by Dr. Patrick Adebola. And then for the first aspect for this afternoon, which will be focusing on the general discussion on the progress report, I'll be calling on Dr. Asrat to lead the discussion. Okay. Okay, so I can continue. Okay, no problem then. So Dr. He just gave me now the go ahead. So generally what we are going to do just an open discussion where each of us and then also the partner can, if there's a reflection or there are some points that need to be there are some points that we need to, some points or challenges that we need to figure out and then have a general discussion in a particular point, we can do that. So the floor is open for general discussion. I can't see anybody raise his hand yet. Yes. Okay. Perfect. Also, if probably you want to have some point or discussion on a particular presentation, feel free to ask, and then in general we will see how to address. And then note that all the presentation we compile them and share it with the participants. So we are still waiting for the first point to be raised. Is it because of the, if we need to do exercise, we can do that, so that probably we try to get ourselves back. There's also coffee in at the entrance for people in the meeting room. People online, probably we can share your own for you. Benin team. Hello, Professor Dancy. Hello, Dr. Amani from Cote d'Ivoire.
Hello. I'm online. Huh?
Okay, so any point from Cote d'Ivoire?
Oh, no particular point. Just maybe to say.
Dr. Amani, please, we cannot hear you very well. Could you please raise your get closer to the mic?
Can you hear me now? Hello.
Yeah, it's okay now.
Okay. I was saying that I didn't have any particular points, except I just wanted to say for the seed propagation, yes, this SAH equipment, it may help our national program to have enough planting material for next year. So it will be very nice if we can get it as soon as possible.
Yeah, feel free.
Thank you very much, Dr. Amani, for that remark. We certainly made arrangements for the materials to get to all our partners. And as I'm speaking now, Pelimo, who is the expert in the SH, is in Benin Republic now, trying to assist our partner in Dasa. So if not because of COVID, we have arranged it in such a way that these materials will have been delivered long time ago.
Okay.
But we're still working on it. We just pray that this issue of COVID died down a little bit. But definitely we have it in our mind. In the next few weeks, we'll try and see how those materials will get to you in Cote d'Ivoire. Thank you very much.
Thank you. Thank you.
Okay, Dr. Amani, thanks so much. I think Jim raised a question. I'm coming. Let me see if I can read it to the public. Okay, Jim was asking, anyway, first he said very nice presentation. And then he would like just to know if there's French people, they are using the YAM base for trial planning, data collection, curation, and analysis. So for this, I think Germa and then Dennis Corner, they are online, so they can give a response on that.
Yes, Dennis Cornet. Thank you for the question. Yeah, so we were not used to use it, but last year we start to exchange with BTI, with Lukas. So he helped us a lot to understand the system, and we only initiated the deposal of our breeding experiments, but this will continue next year, and we will, yes, start the process to use it regularly.
Okay, thanks so much. So since we have finished with Cote d'Ivoire, I will move to Ghana now with Professor Otu. Professor Otu? Hello? Yeah, is there? Is online was?
Yes, I can hear you.
Okay, perfect. So we would like just to hear from you on the discussion. What do you have, and then if you need some aspect to be clarified, so we are listening to you.
Oh, I nothing to nothing to really talk about. That's why I've been quiet. I've followed keenly the discussion. I think we've received our planting material for the regional varietal trials. They are with us. We've already prepared our land. And so we started planting. So I think everything is on course. We have no problem.
Okay, good. So I will move to the second breeding program in Ghana and then call on Dr. Chamba if he's on line. Dr. Chamba?
I'm online.
Okay, perfect. So we would like just to know also the plan for the on-farm and testing material. What is your plan for that on-farm trial?
Well, as part of our plan for the year, we are repeating what we did last year. And so, yes, we have plans for that, as well as the multi-locational trials. We are yet to start because the rains haven't started here in the north, but preparations are going on in terms of seed. So once it rains, we will start land preparation and also planting. But let me just also mention something about the botanical seed. I've been thinking about how one can determine the viability of botanical seed before you do the sprouting. It's an issue that I've been thinking about. How do you know that the botanical seed that you have received is viable and not just an empty capsule that you are planting?
Okay, good. Let me provide an answer, and then a team member also, if possible, they can contribute. Generally, what we are doing in IT here, you have the shape of the seed, and then the thickness of how we can call it. I don't know if the umbra, if we can say that. Depends how very thick it is, then you can have an absorption if it's going to be really viable or not. That is the key. And then by looking at the seed, you look at the embryo. Some of them, they are not really fully developed. You can now disqualify those ones. So we have used that technique for kind of like more than 1,000 seed, and then we came for that conclusion to see the thickness part of the embryo determine the viability of the seed. So you can use that something like visual assessment. Sorry, I did not hear what you said.
No, I said, how do you determine the thickness of it? You know, this is just as thin as paper, you know? And
yes, you just put it between your two fingers, and then you assess it. Some of them, they are very thick, and then from that, you have a very good which expression can we use? It just, I think the thickness between your fingers, so you just assess it that way. Okay.
That for the viable seed, when we mean when you feel the seed, the small seed with between your fingers, you can see it is a little bit hard. But if there's no seed or no embryo, it crashes and you don't see anything inside. So that's how we are just, after crossing, we differentiate viable and then viable seed. So that viability does not mean that it will be 100% germination. So if you want to know about viability based on germination, then you can sample. If you have many hybrid crossbreed seeds, you can take 100 and then see the germination percentage. You can sample it and see germination percentage. And then based on that germination percentage, you can plan for how many seeds to sow in order to get the amount of population you want to start with in your breeding the nursery. So that's how it happens.
Is it okay? Are you okay now, Dr. Chamba?
Yeah, somehow. Okay, we'll talk about it later.
Okay, so another question that me particularly I would like just to know, just last year your team and then complained about the nematode effect. So what's your plan this year? What are you taking into consideration to select your testing? Yeah.
Back to me. Just like myself.
Hello, we can't hear you very well.
Oh, the question is to me.
Yes.
Yes.
Asking you.
Okay. What happened last year was that we changed the site to a field which has been left fallow for almost 10 years. Unfortunately, that field was highly infested with nematodes, as we have seen in the tubers that we have harvested. This year, we are going back to the field that we've been using before last year. We allowed that field to go fallow for 1 year. And so we think that where we have been using previously, we never registered incidents of nematodes. So we will go back to our previous field. And we're hoping that that will resolve that particular issue.
So we don't know if you have a capacity to test nematode the density in the test in the site you are going to use. That
we don't have that capacity. We don't have that capacity. We don't have a nematologist at our institute.
So that we can avoid the, I don't really know if Dr. Asrat can make some suggestions so that we can avoid such a result at the end of the year.
Okay.
Hello.
Hi.
There's Emmanuel from Ghana. Otu. I think we have a nematologist that you can liaise with to assist.
Okay.
Dr. I've forgotten the name, but he was in my office from minutes ago. He is a pathologist, he's working with us, and he can assist. So if you contact him, so I would let him know.
Okay.
And you can also talk to him.
Okay.
Yeah.
Thank you for the information.
Okay.
Okay. Thank you for that suggestion. Okay. Let me see the test if there's a question. In-house. Okay. Okay. There are already some questions that I will read it out, and then the project leader also can comment on that. Following presentation from partners, I'm tempted to link team are working on similar topic. Is it there any clear task description for different team involved in AfricaYam? It will avoid repeating activities, case of genomic analysis, for example. It's from Mundo. Yeah.
Thank you very much for that question. We're really following the project documents, and we assigned tasks to individual partners. And I can tell you, we have synergy, but there's no overlap in terms of activities that are being carried out by each of the partners. We have opportunity to work together in some areas, but there is no repetition or people doing the same thing from by different partners. So yes, there's no duplication in terms of the activities and work assigned to the partners. Thank you very much.
Okay, thank you, Dr. Patrick. We'll go to the next question in the chat. I think it's from Dr. Akinsowe. I think so. Thanks for every research team. You did very good work. Question from consumer acceptance, the rural and urban. Okay, let me read it from here. It's much better for me. Okay, from consumer acceptance, the rural and urban differed in preference. From these results, what breeder can do if his aim is to breed to meet consumer preference, breed for urban or for rural. Yeah. The second aspect of the question is for critical analysis for controlling it rates. I observe that IT and CIRAD work on different it rates. Is it a constraint problem for the project? Probably I can try to answer for the last aspect. For the last aspect, there's no constraint. I think the trait were assigned based on the capacity of each breeding program. Like during the presentation, the weed were assigned to the French people based on what the capacity they have. So it's not really a constraint. Now for the first aspect, I think I will call on Dr. Asrat if he has some point to address. Also for the team. Yes, for rural consumer.
Maybe other breeders from the NAS. Dr. Amani, can you respond to this question? Professor Dancing, Jude.
Hello. Hello.
Yes.
Okay, let me try for this question.
Okay.
Urban market is a new market which is expanding. So I think for future, we need to breed for urban market, but we don't also forget what do you call the rural people who are farmers. So we can breed for both. In some of the aspects, they are linked. So in some aspects, they are linked. Even if we can find some small difference, slight difference among these the two products. So we can breed for urban and rural people. It is possible. And some of the products are the same.
Maybe others?
Thank you. I think I want to subscribe to what my colleague Dr. Mane said. These are different markets, and in line with our product profile development and market segments discriminations. I mean, we need to have a target market when we are breeding. So I don't see what's wrong with targeting urban and rural markets if you have the capacity. So I think, yeah, of course, at some point, some of the threats will overlap, definitely. But if there are specific threats that can fit the niche of urban markets, which is not found in the rural market, then there is need for us to address that as a breeding team that is sensitive to emerging markets. And I feel these brings up opportunities for us as well to evolve our programs in meeting modern demands, kind of. Yeah, thank you.
Thanks so much for your contribution. Professor Happiness, any contribution? Okay, I'm good. So I think we are making progress. Let me check if Professor Dancy is online now so we can. Hello, Professor Dancy. I don't think he's online. Okay. So from
yes,
BPI to...
I am online now.
Okay, you are welcome.
The internet connection is fluctuating so much this morning, so it is difficult for me to follow all the discussion. So I am now connected. So yeah.
Okay, so let me just try to give you just a few feedback on what we are doing currently is on general discussion. And then
yes,
we would like to hear from the partners and then everybody participating to know exactly what is the feeling and then what is probably issue on what can we improve or something like that. And also have a general overview on the different presentation we had so far. So we are listening to you if you have some point to be discussed.
yeah. Thank you very much, Dr. Agree. From my side, I just want to thank everybody for the excellent presentation. For the program coordination, when they send the template to us to fill, I think somebody already raised the situation in the morning. There is no place for the activities other than what is listed. But we came from phase 1 to phase 2. So the activities of phase 1 are there, but there are some other activities concerning, selection of a good genotype that is already ongoing. So next time I suggest that there should be a line where we can put those information. And also it is good to have the template not at the end of the year, but now at the beginning, so that when we are doing the activities, we will be checking and know what is clear to deliver or what is necessary, the line to consider when developing our, our report. That is the only one, comment that I have. If not, I am very happy with what is going on in terms of breeding. And somebody, let me also add this. I think somebody, it was a Dr. Asiedu, I don't know, who raised one situation. Varieties, there are some varieties that are released in some countries. In fact, it stay in that country. So there should be a kind of collaboration between the countries involved in the project, so that a good material found somewhere be shared with the other countries for multiplication, evaluation, and use. It is really important. So if we put effort together, we will go far. That is one thing I really want to highlight. I want to take as an example the regional evaluation samples that was sent to the different countries. I really appreciated that for in Benin, because we have very good genotype insight. let us continue by sharing another type of material like that to add to what we are developing in our different countries. Congratulations to everybody, and that is my impression. Thank you very much.
Okay, thanks so much for your comment, suggestion, and then yeah. So I think in line in collaboration with the project leader and then people involved in AfricaYam project, we try to monitor the different activity and then how to be sure also that every partner is doing what he's supposed to do at a real time. And then the second point you raise also, I don't really know if if in Benin or other country involved in AfricaYam project, the different law in place, if it allows any material released in Nigeria, if it could be also released or freely used in respective countries. So if it's possible, there's no problem. That one can be done easily. So thanks so much for your suggestion. So we check on, oh, sorry, I'm listening to you.
Yeah. It is good what you said, but you know, taking the case of Benin Republic for instance, I did a small survey 1 month ago, and I realized that farmers always import some genotypes from Nigeria. And actually, we have 3 or 4 like that running. I mean, everybody is demanding, requesting the variety, the seed of the varieties, and it is not easy even to find it. Why? Just because they find this very good. So what maybe we refuse to do officially is what the farmer used to do, and we now go to them to sample material from them. I think since the beginning of this project, I've started to, used to say that it is important those type of exchange of good material from country to country. If, for instance, Ghana give 5 bears, Nigeria 5 bears, Benin 5 bears, Cote d'Ivoire 5 bears, and we put it together and we start evaluation as another small activities of our program. I think we will quickly reach a certain number of interesting genotypes to replace what is there now. I'm telling you that what we consider as how do you call it, popular or market varieties, some, although they are still market varieties, our popular are decreasing in terms of production. So we should think about that. That is just a suggestion. Thank you very much. Agree.
Okay, thanks so much, Prof. Your points are well noted, and then we see how we can integrate those aspects in the project. Okay, so let's check on our partners. BTI, hello, Lukas.
Yes, hello.
Okay. Yeah, we would like to hear from you.
Yeah, so I don't have any specific questions to anyone except that I would like to encourage everybody who works with the database to in case of problems, to contact us, and we can resolve issues if they arise. So that's maybe the most important point.
Okay, thanks so much. Our collaborator from France.
Yes, Denis again.
Denis.
Okay, no, not so much to say. I get the point of using YAM base, and from the presentation of Lukas, I'm seeing the advantage of using it. So I get the point of Jim and Lukas that we need to hasten the uploading of our data on this platform. I forget to say that we initiated the process. And the first step was also to make some adding on the YAM curation ontology tools. So we had our traits we had more than 12 traits, quality traits with different methods in order to be able to upload the data later on. So this is an ongoing process, and I'm sure next year we will have results about that and data upload on the platform. Otherwise, I just like to thank also to have had the opportunity to present our results, and yeah, that's it for me. Thank you.
Okay, thanks so much, Dennis. So, other point to be discussed. Okay. To our partners, we would like to hear from you, the on-farm, what are the different procedure you have put in place right now to carry out, and also for probably the RVT, the selection site. How do you select them? And then we would like to know if you consider the different agroecological in your country. So I will call first on Professor Otu, so he will give us a view on those two aspects. So Professor Otu, over to you.
Agree.
Yes.
Yeah, I missed a little bit of it because I thought we were. So if you wouldn't mind bringing me to speed as to exactly what you were talking of, because I just.
Okay. First, for the on-farm activity to be conducted in your breeding program.
Yeah.
We would like to know your reflection, have a reflection on it, and then how you, the procedure you have put in place. And then for the RVT material we just received, how do you select the three locations? And then we would like to know if you consider cover most of the agroecology in your country.
Thank you very much. We do most of our work, the southern Ghana, within the forest savanna transition area. So we have Ijura, Kintampo, Atebubu as the chosen locations for the RVT. And so we wouldn't change it. That's where we do our work. So we wouldn't change it. Of course, with your support, we've designed a trial. We have the layout and everything. So we can follow the protocol for the RVT. The on-farm trial will also try to look at having a minimum of 3 communities apart from the mother trial to see how best we can spread the material within the agroecology and then evaluate them. So seriously, we don't have any bottlenecks as at now. We see everything ready to go. Don't have any complaint. We also know that you are at arm's length if we need any assistance. So we don't have a problem. We appreciate work done so far. Thank you.
Okay.
Thank you, Professor Otu. I don't know whether Glenn is still online. Glenn, are you there?
Yes.
Okay. This question is.
I am.
This question is for you. You know, because of the pandemic, we were not able to fulfill all what we promised to do last year. I want to know if there's any plan for any activity from your side next year, training, visits, or if there's any activity for next year, assuming that we are able to overcome these traveling restrictions. Thank you.
Oh, Glenn?
Oh, was that me you were talking to, Patrick?
Okay. Hello, Glenn.
Yeah, I didn't hear Patrick very well there, but and then somebody else responded on my behalf. I don't know who that was. Is that? Oh, there you are. Come in, Patrick.
Let me repeat my question.
Yeah, I mean, I think this is something we need to discuss. I think there are some possibilities for some training potentially later this year, or we'll have to see how things go with the pandemic. But I definitely think there are some possibilities either for training over in the UK or training in Africa towards the end of the year, maybe. I think traveling backwards and forwards till late summer, I think is going to be a problem potentially. But I definitely think there are some possibilities which we can discuss. Yeah.
Okay, thank you. Talk about it maybe.
Yeah, well, let's discuss it offline. I definitely think there are some things we can do, and certainly, I mean, certainly towards the end of the year and maybe in the beginning part of 2022, I think things should be much more, hopefully, back to normal, whatever that means. You know.
The vaccines are out now, and we seriously envisage that before the end of the year, things will come back to normal. So I'll talk to you outside the meeting.
Yeah, no, that would be really good, Patrick, and I'm really willing to work with you on that and see if we can get some things going. You know, maybe from August onwards, hopefully.
Okay, thank you very much.
Cheers.
Okay, thanks so much, Glenn. So we are back now with our partners with our question. So we have heard from Professor. No. I can speak. Yeah, it's okay.
Hello.
Okay, so I think we have, you are muted. I think we have heard from Professor too. I don't know if people are receiving me.
Hello, I can hear you. I've already spoken. Or you want me to speak again?
No, it's okay. So, Dr. Chamba, it's now your turn. Let's finish with Ghana first.
Yeah, that's right. Thank you very much. With the RVT,
yes,
we are working at 3 locations, and there are 3 different agroecologies for yam production. So our trials will be repeated in those agroecologies for these 2 different RVTs. Then on farm, we adopted the approach of the baby mother trial last year. We worked with about 15 farmers in 2 locations. We want to repeat that and get 2 years information. So that is about the on-farm. But we also plan that we'll be distributing some materials to farmers and 3 agroecologists. So we'll be working on that as well. But let me come back to the RVT. We have done RVT before, but we got the protocol for only 1 year. And I think after 1 year, the partners were moving in different directions with regards to the material. So I will suggest that we should be able to get protocols that will carry us through to the end, to the point where we'll be releasing the materials. So this time around, I want to believe that the protocol will not just be for 1 year, but will also be guided as to what to do the next year and then the year that will follow it, so that things will be uniform across the partners. This is my observation. Thank you.
Okay. Thanks so much for your point. I think it's also very good. And then also from IT, we discuss more with our partners to follow them and see if the target is going to be achieved based on what we really what we need for the project. So I will now go to our Cote d'Ivoire partners. So Dr. Amani, if you are there, back to you. Yep.
Yes, for the on-farm trials and this regional trials, we take into account the agroecological zone. You know, in Cote d'Ivoire, we have 3 agroecological zones for yam growing area. We have the savanna, we have the transition zone, and we have the forest zone. And our trial in these places, for example, Boaké is the transition, Ferke is the savanna, and Ganyua is the forest zone. So we consider all these zones. The third for Dialata will be another zone. We can call it transition zone. So we take into account all these things. The number in the paper Dr. Asrat sent us, he asked us to have at least, I think, 20 or 40 farmers, but cannot be reached because we don't have enough planting material for 40 or 20. So we'll have, I think, 12 or 13 farmers across these regions.
Okay, that's
Thank you.
is okay. We are still expecting also the checks material you want to use for the RVT, for Rotundata and also for Alata. We have received from Ghana, Benin, and then we are expecting your own very soon, so we can share the design of the RVT with you.
Sure, we'll do. I was on a meeting last week and I asked my colleagues to do. They could not. We are going to do it.
Okay, good. Okay, so Professor Dancy, if you are online, is now over to you.
Yes, I'm online. Thank you, Dr. Agri. It's almost the same thing with the other colleagues. In Benin here, we have selected 3 sites. As you are from Benin, you really know the places. We have 3 agroecological zones. The Savannah zone, where we selected the village Tokotoko. And then we have the transition zone, which is Basila area, where we selected the village Talu. And then we have the forest zone, Asaba, within the Bante district. You know, Bante is a forest zone. So we have selected 3 sites 3 sites where we are going to test the regional, to do the RBT. That's the way we selected the villages, and the protocol or the design was recommended by your program, IITA. So we are exactly going to use that one, so that the result with the other countries will be, uniform. That is a very important point. For the on-farm, we also consider agroecological zones, but within the agroecological zone now, we selected the certain number of villages, and 10 farmers from the villages according to the availability of the material. Actually, we are working on the seed available. I will let you know exactly how many farmers will select per village. But actually, we are planning to work with 6 to 10 villages and 5 to 10 farmers within a village. The final number will be given to you as we are now trying to count the seeds that are in a good condition to provide a good result. That is almost all I can say. Yeah.
Okay, thanks so much. So I think we have covered most of our partners, our online partners. So remaining now our in-house partners. So Dr. Jude and Professor Happiness, we would like to hear from you.
Yes, the meeting has been a very rewarding one. The information shared, every partner presented, and we learned from each other. And I think as we go on and things are streamlined, hoping that the COVID-19 pandemic will soon come to an end, we are hoping for a better tomorrow. Because to tell you the truth, online meeting is good, but one-on-one meeting is better, because you see each person physically, you talk one-on-one and ask your questions and so on. So we are hoping that very soon COVID-19 will end. I don't think it will come to the issue of asking each person for COVID-19 vaccine before we enter the plane to travel, and then we'll have opportunity to come together again. So I will just say that every activity that is lined out for the year will be pursued vigorously. I'm encouraging the project manager and Dr. Asrat to send the work plan, the agreement, very quickly. I'm calling Dr. Asrat because I know that my previous work plan, there was no issue of multiplying for demand creation trial in my own work plan. Okay, but then if that is quickly sent across and we know exactly what we ought to do, we will do them. And our senior partner, NRCRI, they are always there to backstop us, and I know that the seeds to get involved both in the MLT and RVT we will get from them. And anything that is outstanding, of course, IITA will supply. So we are ready for action for the year. Thank you very much.
Thanks so much, Professor Happiness. So we'll invite you to speak on the on-farm and then also on the RVT. What are the different actions he's taking? from yes.
Yeah, I think Professor Oselebe has actually represented the Nigerian team. But for the demand creation trial, it's another trial for us. And this is the first time we are going aggressively on farm to validate our release materials with farmers. So for us, it's very sensitive that the farmers are actually going to the field already. So I think efforts are actually being made, materials are ready. So by the time we get back in the next 1 week, farmers should start receiving this material because it's good for them to plant this material as they're planting theirs. And I think for us as an institute, we are poised for that activity. And for the regional variety trial, where we manage, I think, up to 6 trial sites in Nigeria. Then IITA will handle some, probably handle like 2 sites. And for the latter, we have 4 sites for Umudike. That's a huge one. And okay, I don't have much to say. It has been a fruitful deliberation. Though boring at times that you can't feel some of your colleagues, you see them on screen. And throughout the year, the meetings online has been getting so boring. I mean, you have a lot of meetings online, 2, 3, 4 meetings every day. At some point, you lose it. And at times you forget appointments on meetings at times. So it's not really, I think this COVID-19 needs to stop actually for the scientific community, for us to gain back our lives. Yeah, so beyond that, I'd like to thank the project manager as well and Asrat, our lead project personnel in breeding, for his support for the Institute and the Nigerian team. Going forward this year, I'm positive will remain a good year for us as far as young breeding is concerned. So thank you very much.
Yeah. I have one point maybe for the others to reflect, especially the planting material preparation for the next year trial. You see, for those regional variety trials, immediately going from one trial to the other trial using this tubers coming from the trial, once we identified good material that's potentially good for more location testing, we then diverted them to multiplication for 1 year. And from those, we managed to now, I think, share about 18 locations of Rotundata and more than 20 locations for Alata. So if we are decided to move for this regional trial last year, it will maybe not possible even to go for 4 or 5 or 6. So really for the other programs, you know, sometimes we have some materials in the release, some also in the process. Then we also give due time for seed multiplication, especially seed techniques, and also use the other techniques that are developed by IBRC project in order to get quality planting material and combine it with the SCH facility so that at least we get enough sufficient and quality uniform, grown at the same field, the same environment, and we use also whole tuber for our planting than the cut sets, which also cause some problem of uniform sprouting in the field. So maybe if some of you are, if you reflect on this, especially for the 2022 trials, what about your preparation apart from conducting trials? Yeah, the effort I think we need to put or those materials we want to evaluate in 2022 to multiply it. So if we get some reflection on that, I think that will be good. Thank you.
Thanks so much, Dr. Asrat, for your point. So any other point to be discussed? If there's no other points, I will call on Jim if he's online and he's available now to give his thought on reflection and key action point from him. So if he's available now.
Yeah, thank you, Audrey. Can you hear me?
Yes, I can hear you.
Very good. Well, I would like to thank everybody involved in these meetings for a very good meeting under the stress of COVID and Zoom meetings. In particular, I'd like to thank the TAC committee. So thank you. Not sure who's still on, but thank you, Eric and Noel, David, and Carlos. What you have to provide is invaluable. And Patrick, thank you for all the logistics and hard work and for your whole team that stood behind you. I know many at IATA were working to bring this about with the advancement meeting and then the project meeting. So thanks for all your hard work. And the same to all the partners across Africa, as well as in Europe and Japan and wherever your partners are. Thank you so much for the good work. While as we heard from the TAC, there's always opportunity to expand the community. It's already a tremendous community and a great delight to see people and how engaged they are in this important crop. So many thanks to you and your teams, including technicians and administrators, of course, all the scientists and postdocs and students. This is really important work. And second, I'd like to thank you both for what you've done to stay safe in the time of COVID and for all the work that did get done in this time where everything was affected by COVID. So please remember from the point of institutions, even us, that people come first and safety is important, but prioritizing the work so that you do what's most important in those opportunities you have to do work is also important. And it's good to see that there's been prioritization and work has continued and progressed. So one of the things I know in the transition to one CGIR, for those people most engaged in the CGIR, those directly within the CG, there can be a lot of stress. And this question raised, what happens next with funding, can be even top of mind. But I would like to assure you that from our perspective and from the perspective of many other donors, funding will continue. The CG is essential. And yes, there are big transitions, but I wouldn't worry that moving towards the end of funding of this project will mean moving towards the end of funding. It's been a huge encouragement to us to see the Crops to End Hunger coalition of donors come together and begin to push the CG to prioritize breeding as one of the core activities for which it exists. And breeding modernization is part of that. And what you see happening in AfricaYam is related to this larger effort to modernize breeding for the benefit of geographies where poverty and malnutrition are most prevalent. So a high focus, high priority on Africa. And with that has also been a prioritization effort of the donors. And among that, within that, then poverty alleviation has been a strong criterion in prioritizing, which means yam is coming out as a high priority crop. And I believe and hope that this will result in a broader coalition of donors supporting the work of yam breeding. And so I think that's really good news. And many of you who are directly involved in breeding will have heard of and know the Excellence in Breeding program. And one of the concepts that we're discussing a lot with the Excellence in Breeding program is regional breeding networks for crops. And again, it's not set in stone, it's not finalized, but I really expect that this will transition to the EIB and the 1CG funding breeding networks, in which there's money going directly to national programs as well as to CG institutes, with both the expectation and the realization that national partners are key in the success of breeding, and they need to be peer programs where they're fully respected as partners, and recognizing that the CG can't do its work without the national programs, but that there's tremendous benefit to be realized by the national programs in collaborating with the CG institutions. And I don't know how far that will go in terms of defining relationships. It certainly will not be that it's expected the national programs will just serve the CG programs, but they'll be fully recognized peers. By the same token, there also won't be pressure on the national programs to do more than they're ready to do. So if their country isn't fully committed to having a full-fledged breeding program, that's okay. They can be very engaged in being part of the testing network and getting excellent material for their countries that they can validate and test and release and get support from the broader network on. And the flip side of that is, if a country is fully committed to having a full-fledged breeding program from very beginning to very end, no pressure to cut back. It's not that the CG will be doing the breeding and national programs are only testing networks, but the expectation that by working together, and in a small community like this, really you're greatly strengthened by working together, there will be mutual benefit and mutual gain. And so I expect there will be funding for that. There may be some changes. It may be the expectation that national programs will carry their salary burden, and that the CG support will be more for operations, not fully, but to help some of the extra costs of working within a network. And then specialized opportunities such as genotyping, specialized support. And along with that, the CG partner will do more to help train and upgrade and backstop the national programs. So IATA will also have to step up in their training and backstopping role with a network like this. Another question I have is this network as broad as it should be? Or are there other countries that should be brought in? If you look at the Yam Belt, it also includes Togo, it includes Cameroon. Not quite sure about countries further west. And no one talks about Ethiopia, which is, I believe, the #6 yam producing country in Africa, and has some very unique germplasm. Let's see. Does somebody need to step in? Okay, I heard someone else talking. Okay, I'm glad that the TAC brought up the issue of seed for trials and the quality of seed. And I thank Asrat for already taking steps with IATA to have seed production plots. There's a lot more that needs to be learned. And I can imagine in future that there would be differences together with some of the genome work you heard about, that maybe early advancement decisions would be made by genomic prediction. This isn't this year, it isn't even next year, but as you work on on genetic relationships and being able to predict performance, recognize that this can become part of the trialing network. And if you can select, say, 50 or 75 high probability genotypes to advance while they're still clean in the pre-field stage, it could be that those are ones that would be then multiplied in a clean way by SAH or whatever technology and distributed through the network so that you can get broad experience with the same set of clones, and that would become part of what you're doing. So networks and seed quality will be going hand in hand, we hope. Also the tech pushing you, Agre, and the others on these quality control genotyping panels to start to use in all the programs, to start to see where are the issues with admixture, with mistaken identity, so that you can start to identify problem areas to work on, just as all the major seed companies have done in their history. That they all recognize that the first use of DNA markers is for quality control. So with that, those are the things I wanted to bring up. But hopefully you heard the message of reassurance, the full expectation that funding will be there for this program, for not only CG breeding, but hopefully also for breeding networks, including national partners. And also, along with what you heard from David, that please do look for opportunities to bring more scientists into it, more advanced world scientists to look at some of the biggest issues that are holding back breeding, some of the bottlenecks, but also looking for genetic resources, using what you heard from Dr. Terauchi, maybe to guide collections, to think about where to get enough diversity, and think about how to access that diversity in a very thoughtful way to find important traits and bring them into the program intentionally in a way that doesn't slow down the breeding with the elite material, but you bring it into adapted material before bringing into the mainstream of breeding, so that you don't take a step backward even as you try to move forward. So we're excited about what modernization can do to help breeding. And I thank you for all the hard work, for the important partnerships that you fostered and are working with, and look forward to hopefully good results and also best wishes in coming through this difficult period of COVID. So with that, I'd like to thank you and wish you all the best. Thanks.
Okay, thanks so much, Jim, for that wonderful suggestion, comment, and then review of the AfricaYam project. And then we'll be always very happy to listen to your comment and also how to improve what we're doing. Thanks so much once again. So I will call on Dr. Patrick, the project leader now, for the final observation, and then for that point to be discussed if possible. So Dr. Patrick.
Thank you very much, Agro. First, I want to thank Jim for your support always. I want to thank you for interest in YAM and the support you have been giving to us. I know what we both passed through when we were trying to put in the proposal for the second phase. Jim really did wonderfully, and he supported us and made sure that the proposal was successful. So thank you so much for your interest in YAM, and thanks for all the support we've been getting from you. On our part, we promise that the hard work will continue, and we'll make sure that all the objectives of the project is achieved. Thanks also to the TAC members for your time. It's not easy to sit there and listen to us for 2 full days of your time. I know we are all very busy people. So thank you very much for all your suggestions also. We'll take all those into consideration going forward. I also want to thank my colleagues, especially Asrat. In fact, I don't even know how to describe you in terms of the hard work you have put into this project. Thank you very much, Asrat. We had a very good relationship, and I believe it will continue, and it has contributed to most of the success that we have recorded in this project. Thank you very much. You are a very valuable member of the team, and we don't know what we'll do without you. Thank you very much. Agre, thank you very much for all those molecular stuff, and you have also worked so hard on this project. I really appreciate what you are doing and what you still be doing. Thank you very much. I also want to say thank you to our partners, Professor Otu, Professor Dancy. We also have Dr. Chamba. We have Professor Happiness. We also have Dr. Jude Obidiegwu. Thank you very much. Our working relationship has been very smooth. We never had issues with one another, and it has contributed so much to the success of the project so far. So thank you very much. To our international partners in CRI, BTI, IBRC, Dr. Terauchi, GHI, Glenn, CRI, Anu, and Connect, and also to Dr. Lukas. Thank you so much for your support. We also had very good working relationship with you, and I believe this will continue. Once again, I want to thank the YAM team, especially in Ibadan and Abuja. They've done wonderfully well. No issues also. I don't have problems of resolving personal issues or friction among the team members. This is wonderful. We have a good team both in Ibadan and also in Abuja. Thank you, everyone. If you are listening, we really appreciate your contribution and support to the project. For the project support, also we want to thank you, Paulette, Olubumi, and also the logistic officer, Israel. Your support is really, really appreciated. So thank you very much. I believe that you continue to support the project until we get to the end of it. I just have one remark about a possibility of involving maybe not more countries in the project. I think it's a very good suggestion from Jim. We have countries like Togo. I kept on wondering why we jumped Togo, moved to Ghana and Cote d'Ivoire. So, well, maybe it's an issue of funding, or maybe it's okay. So yeah, we're looking forward if the funding allowed. We have countries that are really producing and also eating yams. And I've been receiving a lot of calls and information from some of these countries saying, why are we not involving them in the project, especially from DRC Congo. Received a lot of requests that they will have loved to join us. So maybe going forward, if the situation of funding allows, we can consider those countries, including Ethiopia and Cameroon. Well, that is all what I can say. I thank everyone again And I want to declare the meeting closed. I wish everybody good or happy Easter holiday. Thank you very much, and thanks to everyone.
Thank you. Bye.
Thanks all.
Jim, bye.
Bye, Noel.
Bye.
Bye, everyone. Good job.
Bye from Ghana. Prof, happiness. Yay. Happy Easter.
Cheers, everyone. It's been great to listen to your work.
Yes, so Glenn, nice seeing you all. And from